In today's lab, we will be doing food tests. These tests are done daily to detect the presence of various substances such as fat, sugars, and proteins in the food we consume. In this lab, however, we will be testing for starch, unsaturated fat, and reducing and non-reducing sugars. Starch is the main carbohydrate found in most ground provisions, as well as rice and flour. To test for starch, we simply add a few drops of iodine to the sample, and if starch is present, then we will see a blue-black color change. This change occurs because the iodine bonds with the starch to form a colored complex. The brighter the color change, the greater the amount of starch present. In our lab, we tested potatoes. A few drops of iodine were added, and after a few minutes the potato pieces became black, indicating the presence of starch. Fats are nutrients present in most foods from animals that we consume. Butter, cheese, milk, eggs, and oils are foods that are all sources of fat in our diet. Fat sources that are solid at room temperature are known as saturated fat or solid fat, while those that are liquid at room temperature are known as unsaturated fat or liquid fat. In today's lab, we will be testing specifically for unsaturated fat. The principle behind this fat test is that since unsaturated fat molecules are non-polar and water molecules are polar, they will not mix. To carry out this test, we simply combine a sample of oil with water. If the samples do not form a solution, then unsaturated fat is present. The final test which we will be carrying out is a test for reducing and non-reducing sugars. A reducing sugar is one that contains a OH group on the central atom. This OH group allows this sugar to act as a reducing agent. Monosaccharides such as glucose and fructose are reducing sugars. Non-reducing sugars do not have a OH group attached to the central carbon and therefore are not reducing sugars. Sucrose is a non-reducing sugar. To test for reducing and non-reducing sugars, we will simply use the Benedict solution. We have already prepared samples of glucose and sucrose, which we will add Benedict solution and warm in water. As you can see, the glucose sample changed from blue to green and then to orange, while the sucrose sample did not change.